Okay, so this is the second topic that I want to address today. Okay, again, remember we start by introducing the pipeline. We introduce the scaling. We spoke about the normalization, and you can immediately see that the scaling, it's 99% of the time it's useful. Normalization, it's more tricky. Okay, it might be useful. I saw example of data set in which the normalization was really useful and removed the batch effect and was able to improve the results, but some other times it simply didn't do a good job. So it was not worth doing that. Downsampling, it's something that it's frequently used. Most of the time, downsampling, it's performed to speed up the downstream calculation. So you have your pipeline, you scale, maybe yes or not, you normalize, and then you want to run tSNE or UMAP or FlowSOM or Phenograph or maybe all of them, right? And if you're dealing with like 500,000 of cells, you don't need to downsampling unless you're running the pipeline on a 10 years old computers. Otherwise, you don't need to downsampling. But what about if you want to run the pipeline on 5 millions, on 10 millions, okay? And you don't want to wait like 1 hour, okay? Of course, the running time depends on the machine, but let's say that you don't want to wait that time. You do downsampling. Well, now, the crucial point of downsampling is that the usual type of downsampling that most of the software can run, which are random or interval sampling, are a little bit risky for the following reason. Think about having 100 cells. And in those 100 cells, there is one cell which is very interesting. Okay, it's the holy grail of your research. And you don't really want to lose it, right? So in principle, you say, okay, I want to run my tSNE and UMAP and whatever on the 100 cells, but now we go back to the issue that you don't have enough time. So you need to downsample. And you say, okay, my software allows me to run random or interval sampling. And the best I can do is to sample 10 events out of 100. Now you can already imagine that you need to be very lucky if you want to sample that single rare cells by just sampling 10. Yeah, it might happen, but it might also not happen. And you don't want this risk. And this is why downsampling, it's considered a little bit of bad things, that you have to do it, but you will like to not doing that. Well, this is true for specific type of downsampling, random and interval. It's not generally bad stuff about downsampling, because also in FCS Express, you have access to other type of downsampling that you see called here target and weighted density downsampling, which are specifically developed to enrich in rare events. So still, you have 100 cells, still you want to sample 10, but with these two guys, you can enrich the rare populations, you can increase the chances to sample that specific cell that you are really interested in. So this is just an overview of the different downsampling type. You see here, I always like to mention a little bit about the good and the bad stuff. So when you do downsampling, in general, the good point is that you have a faster downstream calculation. Okay, that's the main advantage. The other good point that you have with target and weighted density is that you can also do this by enriching in rare events, which are the negative sides. If you use random or interval sampling, you might lose rare population. If you do density dependent downsampling, just be aware that after the downsampling, since you are enriching in rare population, you are not going to calculate percentages. Okay, so if you are enriching in rare populations, for example, you have 100 cells, one is the coolest one, you sample 10. And the density dependent downsampling, it's so good that among those 10 cells, it's actually sampling the one that you're interested in. Now you have one cell of interest out of 10. So before, in the original sample, the percentage is the frequency was 1 out of 100. Now it's 1 out of 10, so 10%. So don't calculate percentages after this type of downsampling. But now that you know this, you can already imagine that that's not the idea of this type of downsampling. Density-dependent downsampling are just a way to increase the chances that you sample rare population, and thus to increase the chances to cluster them, to find them, to map them in tSNE, to map them in Phenograph, in UMAP, in TreeMap, and so on. Okay, once you have found the population of interest with tSNE, UMAP, FlowSOM, and whatever, you can phenotyping it. You can understand which is the phenotype of that population, and then you can very easily go back to the original sample and calculate the actual frequency. Okay, now that you know this, you can see which is the real advantage of these two guys. Now, random sampling, it's really intuitive, like it's just random, right? Interval sampling, it's basically random, but it's faster. So if we go back to the example of the 100 cells and you want to sample 10, interval sampling will sample cell number 1, 11, 21, 31, 41. Usually you use interval sampling and not random sampling because it's faster. Now you might say, how much faster can be the interval sampling? Well, if you're dealing with 10 million data points, even if you can save like 30 seconds, it's a good it's a good point, right? Density dependent downsampling, I want to go a little bit more in depth with that. As the name tells you, they are based on density. Okay, they are based on a measurement of how many cells you have in a specific region. This is why they are able to enrich in rare populations. We will spend a few words in the next slide. Overall, remember, when you change the type of downsampling, when you change the size, so the number of events that you are downsampling, you are changing the data set, right? So don't be surprised if when you sample like 100,000 cells out of 1 million, 500,000 cells out of 1 million, or 10 cells out of 1 million, your tSNE plots, your UMAP plots will look different, okay? Because the tSNE plot and the UMAP plot and the FlowSOM plot will be calculating, will do the calculation on a different set of cells. So density dependent downsampling. Why I want to spend time on these two guys? Because they're really interesting, first of all, because they are poorly known, and because like interval and random sampling are really like self-explanatory. So density dependent downsampling, you see basically definition at the top. Algorithms are able to define whether to keep a data point, whether to sample a data point from the data set based on the density of data points around it. What does it mean? So let's have a look. This is the same picture we used yesterday, right? This is the unscaled data. And you will see why I'm using again that picture. So we calculate the density of data points around this guy, which is the red one. So how many data points we see? Like 5. Okay, 5. How many data points we see here? Like 1. How many data points we see here? Like 2 or 3. Okay, so let's try with a data points here. How many data points there might be? Now, just by eyes, they might be like 10 or 100 or 1,000, but anyway, much more. Let's say 100. How many data points here in the close to the origin? I don't know, 100, 1,000, 1 million, but anyway, much more neighbors around this data point. So now you can see what the density dependent downsampling does. So if you have for each dot, each data point of the data set, the density dependent downsampling, it's deciding whether or not to keep it. And if the density is low, like the red, the blue, and the green, the algorithm will decide to keep it. If the data point has a lot of neighbors around it, the algorithm will likely decide to not keep it. Okay. Now, it's not like yes or no. It's a matter of, let's say, a ratio. So the chances to keep it decrease with the increasing of the neighbors. And now you can see why you need, if you run density dependent downsampling, you need to run it on scale data. Remember what we said yesterday? It's not only a matter of which steps you add to your pipeline, it's also a matter of the order that you run those steps. So for an interval downsampling or a random downsampling, it doesn't really matter whether you run the downsampling before or after the scaling. For a density dependent downsampling, it does. And now you can clearly see that if you calculate the number of neighbors here, this might be like 10 or 20. Okay, the number of neighbors here, this might be 20. The number of neighbors here, this is like 4. Okay, so if you look at the plots on the left, the red, the blue, and the green, they all seems to be rare events, but they are not. Only the blue one. And you can clearly see this only if you work with the scale data. Okay? Okay. So this is a fake example. Okay, this is not a real flow cytometry data. Then I will show you real flow cytometry data. This is a fake example. It's a very simple, and I think it will help you in taking home the message. So what I've done here was creating a fake data set with 7 different population decreasing frequency. Okay, what you see on the left is the full data set. It's 161,000 cells, data points, 7 population. This guy contains 100,000, 50,000, 10,000, down to 20. So the red one only contained 20. This is 0.01%. Okay, I said I wanted to stress a little bit the system. So I said, okay, I'm really in a hurry. I just want to sample 5,000 events with either interval sampling or density dependent downsampling. So interval sampling is just picking cells like the first one, 21st, 41st, and whatever, up to 5,000. Okay, so it's basically a random sampling because events are acquired randomly from the tube, from the plastic tube on the instrument. And you can clearly see that with interval sampling, and the same thing will be with random sampling, the frequency are maintained. Okay, so this is 61, this is 61, this is 30, 31, basically 31, 6, 6, 0, 6, 0, 6, and so on. But you see that the frequency is maintained, but the risk is that you are not sampling enough the rare population, which are usually the one you're interested in. So if you are in this scenario and you ask an algorithm to find out like clusters, or maybe you ask tSNE to create some islands, it will be very hard for the algorithm to understand that this is a population and this is another population and this is another population. Probably those two will be considered as a unique, very rare, sparse population because there is not enough information. Okay, you have a lot of information here, 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 but you have not enough information here. So if you do the same sampling with the same size, but using the density dependent downsampling, the algorithm, it's calculating how many neighbors each dot has in this data set. And you can immediately imagine that most of the neighbors here, most of the data points in this population has a lot of neighbors, like 100,000, actually like 99,999, blah, blah, blah. Here, each data point has very few neighbors. So those will be preferentially sampled. those will be preferentially, but a little bit less than the red one sampled, and so on. So at the end, you have something like this. So looks, out of 20 red cells, you have 14. So out of 100, you have 17 instead of 1 and 2. Out of 500, instead of 13, you have 34. So this is a little increase because it's like 13 cells more for the red one, but it's a huge improvement in terms of information because 14 out of 20, it's 17% of that population. 1 out of 20, okay, it's 5%. And you see the price you're paying to enrich those population, it's not that high. You see here, instead of 3,000, almost 3,100, you're basically sampling the same. Instead of 1,500, you are basically sampling like 60 less. Instead of 300, it's 288. So you're losing very little information here, but you're gaining a lot of information here. Sorry, losing a little bit of information here, but gaining a lot of information here. And this means that tSNE and UMAP and FlowSOM and whatever will access more information. So this is what I've done. This is the same data set, and those are the same cells as before. This is tSNE on the intervally sample data set. This is tSNE on the weighted density sample data set. And the game I was playing, it's let's see whether tSNE can find out that those two populations are two different populations. So I was expecting to find two different islands, one red and one pink. Okay, that was my hope. But you see, this is 0.01 and this is 0.006. So they are very, not very, but quite rare. Now, I started with like 10,000 and I couldn't see any separation for those two. 20,000, 30,000, 40,000, 48,000, I cannot still see the separation. So I can tell you, because I know I do backgating, that these little islands here, now it's colored because I did backgating, but when you are analyzing a 40 color experiment, you don't know which is which. You run tSNE because you want to understand which is which, right? So you just see an island, you think this is a homogeneous population, and you will find out that this is a two population merged together in a single island only after a lot of plots. Okay, a lot of backgating, a lot of 2D plots to find out that this island contains both the pink and the green. And this is 48,000 out of 160. 30% of the events. Of course, if you keep increasing, like if you go higher, maybe you need to reach the top, like 161,620. So you know downsampling, at some point you will probably be able to see two islands. Okay, but the price you need to pay is that you need to wait longer. With the density dependent downsample, I was able to run, in this case, the same amount of cells I try, and you can clearly see the two islands. But the cool point is that I went into the other direction. So from 30%, I went down. Instead of 48,000, I tried with 40 and it was working. 20, it was working. And I found out that even if you sample with this experiment, with this fake file, 5% of events, you will still see this good separation. You will still see an island for the green and an island for the pink. So at the end, like this is tSNE. So tSNE, it's like between 2 and 10 times faster in FCS Express than in other software. But if you also add a step like this, in which you can downsample but enrich and repopulation, it can be like, I don't know, 50 times faster because you have like 13 times faster only thanks to the downsampling. Okay, so there is a question, will you get the same outcome from every run of tSNE for rare population? So tSNE, UMAP, FlowSOM, and whatever, those are based on some random initialization. So it's frequently that you hear that if you run tSNE on the same data set with the same setting twice, you get two different results. So this is true, but it's not completely true, meaning that in informatics, nothing is really random. Okay, so there are tricks in which you can ask the computer to find out 100,000 random numbers, and then after a month, you can ask 100,000 random numbers and receiving the same 100,000 numbers than the month before. So in FCS Express, we put in place tricks like this so that you will always get the same results if you run tSNE, UMAP, or whatever, multiple times with the same setting on the same data set. Of course, if you change the data set, like if you change the gates, if you change the compensation, the scaling, and whatever, you will have a different tSNE, but that's another story. So this was a fake file, and you might say, well, but this is a fake file, and it's not a real file. Is this working also with real file? This is another real file. So it's a very basic file. So those are two markers, and my question was, I want to run tSNE. This is like a 20,000 events. It's not really, but please take home the message. This might be 20,000 in this case. You might have 20 millions, right? So can I run a tSNE and find two islands, at least two, for those two populations, those two, let's say, a rare population, okay, this one and this one. Okay, so I did it. On the left, you see tSNE. On the whole data set, you see it's a very small data set. It's less than 30,000. And I had this. I search a little bit within the tSNE map. Here you have like one island. Here there is another island. This is another island, very small island. Those are very separated. Now I enlarged the dots a little bit, so it seems that those two are merged, but actually they are clearly separated. And after a few backgating, I found out that these two populations are actually three. Okay, you see one island here, one island here, one island here. The reason is because these, which I thought to be a single population, it's actually containing cells that are negative and positive for another marker. We can call it marker 3. So actually I was surprised, and I found out that I actually have 3 populations in that region. And then I ran tSNE on an interval downsampled 2000. So it's like 6-7% of the data set. So again, I wanted to stress the system. And you see, I have so few events sampled that tSNE has not enough information to properly map these events. Okay, it doesn't understand that those are actually 3 populations. So I just found an island here, but only because I was really searching for it, which is pretty close to these other islands. So probably if I was not searching it, I will never find it. And it seems to be an homogeneous island. And then I've done the same on a density dependent downsampling. You see, I have many more events here than what I have here. And indeed, tSNE found 1, 2, and 3 islands. Okay, you see same amount of events, same running time, basically, okay, because we're running tSNE on 2000, but definitely higher chances to find rare population. So am I telling you always run density dependent downsampling? No. Most of the time, probably you will not even need to run a downsampling. Again, if you have like tSNE, if you want to run like tSNE on like alpha million of cells, you can run it without any downsampling. If you have a good desktop, you can probably like run it also on 1 million. Just keep it into the drawer, into the back of your minds. You have option to go farther than the classical random and interval sampling that might be very useful if you're doing something really hard, like 10, 20, 30 millions of events and you want to downsample. And it's not really rare that you are actually going up so high with the number of events, because when you're running high dimensional data analysis, after the pre-processing, you're going to merge data. So maybe you have like 100 files and you have like 100,000 cells each. So at the end, you have 10 million. So it's really frequent. Again, just remember that there are pros and cons about the downsampling, so keep always in mind. So before concluding, actually, I want to conclude by summarizing everything and by like putting together all what we have learned. So let me go back to the layout that we were using yesterday. Okay, we have a layout, few plots, few gates. We have a population of interest, which is the cells here. It can be anything you want. You go to tools, you go to transformations, you go to pipeline. Okay, and then you start adding steps, right? So we know that most of the time the first one is the scaling. Okay, and then you might want to add, maybe you want to add a normalization or a 0-1 scaling. This is up to you. Maybe you want to add a downsampling. Okay, it might be an interval downsampling. You can also, for example, add a gate step where you want to put it. Remember, usually you want to put it here before the scaling, because if you're using the automatic scaling, you want the automatic scaling to find the best transition point on the population of interest. What else? What about if you want to use a density-dependent downsampling instead of the interval downsampling? So let's add, for example, this one. Is it in the right position? Yes, because I want to run it on scaled data. Remember, we are calculating. So here I'm going to check all the fluorescence I want. Okay, probably all. And this weighted density downsampling will be run on the scale data. Okay, right? All of them. So do not put this upstream, because otherwise the density dependent downsampling will not access the scale data. If you're running the interval or the random sampling, then you can put it upstream. That's fine, because interval and random downsampling do not rely on distances. Okay, this is a good example of thinking that you need to do when deciding where to put the let's put it back, where to put the steps. What about if you want to add cleaning step? So we have FlowAI, FlowCat. We didn't go into much detail. We didn't go into any detail about FlowAI and FlowCat, but FlowAI, it's supposed to be run on unscaled, uncompensated data. So it requires a little bit of more modification to the pipeline. FlowCat, it's easier with this regard. So it's supposed to be run on compensated and scaled data. Okay, so where you want to put it? Where? Before the downsampling, because it's supposed to be run on the whole data set. And actually, it should be also upstream the gating step. So probably I should do these and these. So I'm scaling the whole data set. I'm cleaning the scaled whole data set. And then I'm downsampling the gate downsampling. And then I'm doing the weighted density downsampling, right? So once you have done these, once you have defined your pipeline, this is just an example of pre-processing pipeline. Okay, again, you might have different scaling step if you want to scale different parameters in a different way. You might have, instead of FlowCat, you might have FlowAI. You might have 0-1 scaling or normalization or none of them. Anyway, once you've done this, the next step is saving a copy of your data. You see here I have 4 data files. You might have 400. Okay, save a copy of your data that will be a scaled, cleaned, gated, and downsampled copy. Okay, you can do this using batch export under data batch export. Okay, you can find this in the manual. What you will end up with will be a copy for those 4 files or for your 400. That will be, again, the scaled, cleaned, downsampled, and gated and downsampled copy of your dataset. So for every file, you will have, for example, 100,000 rare enriched events within the cells population, cleaned and scaled. And those are the files that you're actually going to merge. Those are the files that are you actually going to run tSNE on, okay? Or UMAP or FlowSOM. So this is the entire story of the pre-processing. It's really important. As it always happens basically in science, garbage in, garbage out. So take your time to plan the experiment, take your time to plan the pre-processings. And again, there is much more to tell about all the algorithms, okay? tSNE, UMAP, FlowSOM, phenograph, whatever. We have a lot of material already present on our website, both as a video material and also as textual material in the manual. So just want to summarize, iDimensional doesn't do any magics, unfortunately. You are always the smarter guy in the room, so always be critics when you look at the result that the computer gives you. Take your time, okay, and don't be in a hurry. Okay, take your time to learn, to read the original paper of the algorithms, read the manual of FCS Express, re-watch the webinars that we have. Really, I think it's good to say that you need to enter in a state of mind in which an high-dimensional data analysis experiment, it's like an experiment that you run on the bench. So you need to plan it, you need to spend some time to create your pipeline. It's like creating a new protocol from scratch. And always remember, data preprocessing, it's really important. So I think we have done. If you have any question, we still have like few minutes, so I will be super happy to answer. Otherwise, just keep in touch. We're going to release many more features in the short term, so we will be happy to help you in getting started with them. Okay, so sounds like there are no more questions. I hope it was useful. I hope you will be super excited tomorrow when you're going, or today when you're going back to your data. So to put your hand in the data and to run some pre-processing step. So many thanks for your patience, for having listened to me for like 2 hours between yesterday and today. So it was really a pleasure. Thanks to all of you. Bye.